Hello, students. Have you ever wondered how your body knows exactly when to produce insulin or when to repair a cut on your skin? Well, all of that is controlled by a fascinating process called gene expression. Gene expression is the process by which our cells use the information stored in our genes to produce the proteins that make us who we are. Just like a factory that only produces goods when they're needed, our cells only make proteins when they're required, and they do so by reading and interpreting their DNA. This finely tuned process allows cells to respond to changes in their environment, ensuring that our bodies stay balanced and healthy. Now, imagine that all of the genes in your body are turned on at once. Chaos. So, our cells carefully regulate which genes are turned on and when. That's exactly what we'll dive into today: how gene expression is regulated at various levels to maintain balance in your body. Now that we know gene expression is important, let's explore how it's regulated at multiple levels in the cell. First, we have transcriptional regulation, which controls the production of RNA from DNA. This is the very first step in gene expression. But the regulation doesn't stop there. Next, we have processing of mRNA. After RNA is made, it undergoes modifications before it can leave the nucleus and be used for protein synthesis. Think of this as editing a manuscript before printing it. Then, transport of mRNA comes into play. Even if the mRNA is ready, it has to travel from the nucleus to the cytoplasm where proteins are made. This process is like sending a letter through a complex postal system. Finally, we have translational regulation, which controls how efficiently mRNA is translated into proteins. Each of these steps ensures that only the proteins needed at the right time are produced, just like a well-run factory. Let's turn our attention to prokaryotes, like bacteria, and how they regulate gene expression. In prokaryotes, the control of gene expression mainly happens at the transcriptional level. Imagine you're a bacterium in a constantly changing environment. To survive, you need to express the right genes at the right time, and that's where the Operon model comes in. Operons are groups of genes that are regulated together, making gene expression in bacteria much more efficient. One classic example is the lac operon in E. coli, which controls the metabolism of lactose. When lactose is present, the operon is turned on to produce the enzymes that break down lactose. It's a beautiful system of control, like a light switch turning on and off as needed. Let's take a deeper dive into the lac operon and see how it works. In the lac operon, there are three main genes that work together to digest lactose. But these genes aren't always active. They're controlled by a repressor protein that binds to the operator region and prevents gene expression when lactose isn't around. But here's the fascinating part: when lactose is present, it binds to the repressor and inactivates it. This allows the RNA polymerase to start transcribing the genes, and the cell begins producing the necessary enzymes to break down lactose. This is a great example of negative regulation, where a repressor protein prevents gene expression and an inducer, lactose, turns the genes on. Now, let's connect this to real life. Just like how we don't want our cells to waste energy by producing unnecessary enzymes, many of our enzymes are produced only when needed. This careful control ensures efficiency in our body's functioning, just like a factory that only runs machines when the product is needed. Finally, regulation in other systems. Similar mechanisms of regulation occur in other systems, like the tryptophan operon. Here, tryptophan acts as a corepressor, turning off its own production once it reaches sufficient levels. This shows how feedback loops are essential for balancing gene expression across different biological processes. Let's wrap up with a recap on gene expression regulation and its profound importance in biology. First, summary of key points. gene expression regulation is essential for maintaining proper cellular function, ensuring proper organismal development, and helping organisms adapt to changes in their environment. It's like the instruction manual that keeps everything running smoothly. From prokaryotes to eukaryotes, in prokaryotes, gene expression is mainly regulated at the transcriptional level, helping these simple organisms respond quickly to changes. On the other hand, eukaryotes, with their more complex structure, have multi-layered regulatory mechanisms managing gene expression at various stages like transcription, translation, and post-translation. Importance in biology. By understanding gene regulation, we can grasp how cells respond to stimuli and adapt to changes. It's crucial for ensuring that biological processes happen efficiently and at the right time. And the role in disease prevention, gene regulation also plays a critical role in preventing diseases. For instance, when gene expression goes awry, like in cancer, it can lead to uncontrolled growth and abnormal cell behavior. Proper regulation helps prevent these conditions by keeping gene activity in check. See you in the next lesson, where we'll dive into more fascinating biological systems. Until then, remember, biology is all around you, happening every moment.